Hello, everyone, and welcome to the 12th installment in our webinar series titled Cell Biology Scandinavia. And welcome to this lunch lecture, where we've invited Dr. Lorena Arranz to present her lecture titled Endogenous IL-1 receptor antagonist restricts healthy and malignant myeloproliferation. My name is Daniel. I will be representing the hosts of this webinar. And I will start off by giving a quick introduction to who we are. And then after I've presented just a few slides, I will hand over the word to Lorena, who will give her lecture, which you are all here for. After the event, we can change the webinar into a meeting where you will all be able to show yourselves and ask any relevant questions you may have to Lorena directly. Of course, there's also the possibility of writing down any questions you may have. So I represent MilMedTech. We're a Swedish company that's also active in Norway. We supply and install and maintain both standard but also quite complex and advanced laboratory equipment. We are a general agent to many different manufacturers worldwide, including our co-host Baker for precision cell culture equipment, but also various types of fields such as sterilization and safety cabinets and microscopy, etc. My job is to tell you that I think you should all culture your cells at physiological conditions and ask yourselves the following two questions. Could a deviation from physiological conditions affect your results? And what about physiological oxygen concentrations? As we all know, we have different oxygen concentrations in different cell types and tissue types. And we need, of course, to mimic the natural conditions of our cells that we culture. A system that will allow you to do this is the in vivo from Baker, which is essentially a glove box meets incubator. It allows you to work in a precision-controlled environment for the parameters CO2, oxygen, humidity, and temperature. You never break this environment based on the glove port system that you enter into a completely controlled and closed system. This is ideal for demanding cell culture, such as hypoxia research, cancer and stem cell research. If you're in Sweden and Norway, you can read more about this on our website, milmedtech.se, or of course, if you're outside of Sweden, you're more than welcome to look at Baker's website directly. That is bakerco.com. So also, this time I was not joined with our co-presenter, since she's traveling currently, but I will just say a few words about Baker. Baker is a joint business between the original Baker business, Baker Ruskin, or Ruskin Technologies, and also Clean Air, bringing together different types of cell culture and safety technologies under one umbrella. They help us with arranging this event, of course, but they also host, their focus is hypoxia quite a bit, and they host an event called Hypox EU, which is a quarterly online virtual seminar series dealing with all matters hypoxia, and it's called Hypox EU. However, they also arrange live events now and then, and these have been very popular and attracted hundreds of visitors. These events are organized together with a scientific committee, including many big names in the field, including, for example, Chris Pugh. And we also know when the next live event in this field is going to be, which is going to be in Dresden in Germany, June the 9th to 12th. And this will be next year, of course. All right. A very short introduction to us and what we do. If we can help in any way with precision cell culture, we're at your disposal. But now I will hand over the reins to Lorena, who will share her presentation with you and present her research that was also published in Nature Communications. So right now we're going to wait for her to share her presentation, and she will show up shortly. Hi, Lorena. Now we can see your presentation window instead of a presenter window instead of the. If you can end presenter mode, perhaps, for your lecture. There we are. All right. I'll hand over everything to you, and don't forget to turn on your microphone.
So now we are all set. Good afternoon and welcome to this lunch seminar from the University of Tromsø. It's really a pleasure to be here today. So thank you, Daniel, for the invitation. I'm really happy to participate in this seminar series of cell biology Scandinavia, aiming at giving visibility to the work performed in the Nordic regions here and beyond. And the work that I would like to share with you today was published earlier this year in Nature Communications, and it entitles Endogenous IL receptor antagonist represses healthy and malignant myeloproliferation. Many of you in the audience may be familiar with the role of inflammation in cancer, and this is a story about anti-inflammation. So how the defective resolution of interleukin-1 beta-driven inflammation may underlie, in this case, pre-leukemic lesion and acute myeloleukemia progression. But let me get started with an introduction to these very powerful cell types that are the adult stem cells. Adult stem cells are defined by their unique functional properties. They are unspecialized, which means that they show a low differentiation profile, but high potential to differentiate into the cell lineages that form the tissue where they reside. They are also dormant. This property is also called quiescence, and it means that the adult stem cell is mainly kept in a resting status, is not very active. And they also have the capacity to self-renew, which means that when the stem cell divides, it's able to give rise to at least one daughter cell that is identical to the stem cell. This property is achieved through either symmetric cell division, when the stem cell gives rise to two daughter cells that are identical to the stem cell, or through asymmetric cell division, when one daughter cell is identical to the stem cell and another daughter cell will follow the differentiation path. This is the process of hematopoiesis, that is the formation of mature, highly specialized blood cells from the hematopoietic stem cell here. And for decades, the hematopoietic system has been taken as a model or a paradigm of hierarchical tissue organization, where the hematopoietic stem cell stands at the top apex of the tree with a low differentiation profile, but high potential to differentiate into all blood cell lineages, and also a high self-renewing capacity that is lost very quickly along the differentiation path. However, from the year 2005, and in particular 2015, with the development of the techniques used for the study of these cells, in particular single-cell transcriptomics and single-cell transplantation, it was suggested that certain branches, certain immature branches of the tree, the multipotent progenitors here, they actually show differentiation bias. However, hematopoiesis was also suggested to be more flexible, and these biased multipotent progenitors are able to give rise to cell lineages that are not the major progeny, all depending on the organism requirements. And today we know that these properties of bias, differentiation, but ability to differentiate into lineages that are not the major progeny apply to all branches of the tree, including the most primitive hematopoietic stem cell. I also would like to give you an example of what quiescence means for the adult hematopoietic stem cell. And in the right panel, you can see how one of these cells divides maximum 3 times per year. However, this is the amount of yearly mature cells produced, more than 100 trillion cells. So this means that this amplified pool of progenitors here are, in fact, in charge of steady-state hematopoiesis, and they will also become relevant in the context of malignancy. So we have a hematopoietic stem cell that is very powerful, but mainly kept in a resting condition. How is this performed? So there are obviously 2 intrinsic factors that contribute, and those are related to genetic and epigenetics. But there are also extrinsic factors that are going to control the behavior of the stem cell, and those are related to the stem cell niche. The stem cell niche is the microenvironment that surrounds the stem cell and that regulates its function, fate, and numbers. Stem cell niches are very complex and dynamic entities. They always show unique tissue-specific characteristics, but there are also always characteristics that are common to all of them. And you can see them here in this illustration. There are always bidirectional communication between the stem cell and the cellular components of the stem cell niche. This communication can also be performed through soluble factors like cytokines, chemokines, and growth factors. The processes of inflammation and scarring take part in the dynamic nature of the niche. The extracellular matrix provides structure, transduces signals through integris, and it's also a storage for soluble factors. There are also physical factors that contribute. For example, in the case of the hematopoietic stem cell niche, there is the blood pressure. And usually niches are hypoxic, and they exert a very fine-tuned metabolic control of the stem cell that is required to maintain its functions of quiescence and self-renewal. The process of blood cell production takes place in the bone marrow, where the hematopoietic stem cell resides. And the hematopoietic stem cell niche in the bone marrow is going to be closely associated to the vasculature. The majority of the stem cells are in direct contact with sinusoids, where they are in direct contact with endothelial cells and with mesenchymal stromal cells that can be identified through different markers. These 2 cell types, the endothelial cell and the mesenchymal stromal cell, are great producers of 2 factors that are CXCL12 and stem cell factor that are absolutely required for the maintenance of the hematopoietic stem cell in the bone marrow. A smaller fraction of these cells is going to be associated to arterioles, where again they are in direct contact with endothelial cells, with mesenchymal stromal cells, and with periarteriolar cells. Traditionally, the most primitive hematopoietic stem cells have been associated to sinusoids, but the group of Paul Frenet also found these cells closely associated to arterioles. And just to give you a glimpse over the complexity of the hematopoietic stem cell niche, there are many other cell types that regulate the function of the hematopoietic stem cell, and these include both hematopoietic and non-hematopoietic cells, like osteoclasts, T regulatory cells, macrophages, adipocytes, neutrophils, megakaryocytes, Schwann cells, and fibers, neural fibers. There are also other soluble factors that contribute, including TGF-beta, CXCL4, GCSF, GM-CSF, and several inflammatory cytokines like interleukin-1 beta. This is the model of sympathetic regulation of the hematopoietic stem cell, where the light input is transduced into the bone marrow, where the fiber releases noradrenaline. Noradrenaline acts through beta-3 adrenergic receptors on the mesenchymal stromal cell, which then leads to a rapid decrease in CXCL12. And as a consequence of this, the hematopoietic stem cell will egress into circulation, and there will also be a reduced survival and quiescence of these cells in the bone marrow. With aging, the unique property of the stem cell, that it's the self-renewal, is reduced over our lifetime. And this is going to be due to direct accumulation of mutations, but also to an impaired stem cell niche function. As a consequence of this, there will be impaired tissue repair. There will also be a generation of age-related diseases, and this will include cancer. And this may seem paradoxical, but sometimes these mutations and the alterations in the stem cell niche are going to induce an increase in the self-renewal of the cells. This is the World Health Organization classification for myeloid neoplasms and acute myeloid leukemia, where the diseases are classified according to the outcome, but this outcome is in fact related or dependent on the hematopoietic stem cell functions that are affected. So the most aggressive is acute myeloid leukemia. Here we are dealing with survival rates lower than 5% in the older patients, older than 65 years. And what happens to the leukemia stem cells here is that they show an increased survival, proliferation, or self-renewal, and also a differentiation block. So it's very aggressive. Then there is the myelodysplastic syndromes, and they are less aggressive than acute myeloid leukemia, but they can derive in secondary acute myeloid leukemia. In this case, what happens is that the stem cell, excuse me, it's reduced in its survival, proliferation, or self-renewal. Sorry. And then the differentiation is also reduced. So these syndromes are also called bone marrow failure syndromes for obvious reasons. And the more benign are myeloproliferative neoplasms. In this case, what happens to the stem cell is that it shows an increased survival, proliferation, or self-renewal, but the differentiation is not affected. During my postdoctoral studies at CNIC in Madrid, we made a significant contribution into the role of the stem cell niche in the pathogenesis of myeloproliferative neoplasms. And we proposed a model in which the mutant hematopoietic stem cells produced interleukin-1 beta, which will damage the niche at the neuroglial level. Both features together, this IL-1 beta-driven inflammation plus the neuropathy, drive mesenchymal stem cell apoptosis, in this case identified through the marker nestin, which then will allow the expansion of the mutant cells. And eventually the fibrosis and osteosclerosis of the bone marrow contributed by other types of mesenchymal stromal cells. And this sequence of events is in fact required for the pathogenesis of myeloproliferative neoplasms, because rescue of the fiber blocks the progression of the disease, and also the sympathetic nervous system compensation on the mesenchymal stromal cell prevents the apoptosis of these cells and thereby controls the expansion of the mutant cells. Other authors have found that interleukin-1 beta has no role on steady-state hematopoiesis because deletion of the receptor has no effect on blood cell production. IL-1 beta-induced myeloid priming of hematopoietic stem cells becomes relevant in the context of injury or infection, and accumulated evidence, including our previous work, show that enhanced components of interleukin-1 beta signaling pathway contribute to hematological malignancies. Interleukin-1 beta is the activator of the pathway. It's the inflammatory cytokine. And interleukin-1 receptor antagonist is the repressor of the pathway. So it's considered a very powerful anti-inflammatory cytokine. Here we studied the role of this cytokine of interleukin-1 receptor antagonist in both healthy and malignant myeloproliferation. And we found that low expression of interleukin-1 receptor antagonist is a marker for reduced survival in acute myeloid leukemia patients, and particularly in the most differentiated phenotypes M4 and 5. Low expression of interleukin-1 receptor antagonist is frequent in acute myeloid leukemia patients, and particularly in the lower differentiation profiles M0 and M3, as shown in our smaller Tromsø cohort, but also in a bigger cohort of publicly available patients. As a surrogate of the activation of interleukin-1 beta signaling pathway, we studied the direct translocation of nuclear factor kappa beta in the hematopoietic progenitor cells of both acute myeloid leukemia patients and healthy individuals. And as you can see in these representative images from the ABNIS imaging flow cytometer, we found an increased translocation of nuclear factor kappa B in patients compared to controls. In both treatment or boost of interleukin-1 receptor antagonist as well as blockade of interleukin-1 beta have a therapeutic potential for acute myeloid leukemia patients in xenograft models, because both these strategies, by treating the mice with these drugs, reduce the leukemic output. So to study the role of interleukin-1 receptor antagonist in healthy steady-state hematopoiesis, we used the knockout mouse strain. And we found that these mice displayed a higher cellularity in the bone marrow, differentiation bias into the myeloid lineage with impairment of B cell development, and reduced numbers of hematopoietic stem cells and multipotent progenitors identified as MPP5 with expansion of the MPP2 population. And the myeloid bias that we see in the antagonist knockout mice is due to the increased levels of interleukin-1 beta, because it is rescued by treatment of the mice with a monoclonal antibody against this cytokine. We also performed bulk RNA sequencing of the different hematopoietic stem and progenitor cells, in this case identified as long-term, short-term, and multipotent progenitors. And we found that the transcriptional activation of interleukin-1 beta-induced myeloid differentiation pathways is at least dependent, partially dependent on NF-kappa beta activation, because this myeloid bias is rescued by treatment of the mice with bortezomib, that is a proteasome inhibitor that prevents NF-kappa beta activation. And in this slide, you can see the main cell types contributing to interleukin-1 beta and the antagonist, as well as the proportions in the bone marrow of wild-type mice and changes with the deletion of the antagonist. All the values are referenced to the wild-type hematopoietic stem cell and progenitor cells. So you can see that the enhanced expression of interleukin-1 beta in the antagonist knockout mice is going to be explained on the grounds of the expanded myeloid compartment, together with sustained engagement for an IL-1 beta positive feedback loop in hematopoietic stem and progenitor cells and in mesenchymal stromal cells. So we wanted to give more insights into the sources of interleukin-1 beta and the antagonist in the bone marrow. And so we performed single-cell RNA sequencing and RNA fish. Here you can see the results for the myeloid compartment, which showed that the most abundant and also the highest expressing cells within of these two cytokines within the myeloid compartment are neutrophils and monocytes. And these cytokine expressing neutrophils and monocytes, they express mainly one or the other cytokine, with a few exceptions, particularly within the most mature G4 neutrophils, which express both cytokines together. Here you can see the hematopoietic stem and progenitor cell compartment. The levels of interleukin-1 beta and the antagonist were not detectable by single-cell RNA sequencing, but with RNA fish, we were able to observe that the highest expressing cells of interleukin-1 beta are in fact the long-term hematopoietic stem cells, followed by the short-term and the multipotent progenitors. And we were also able to see that some of the interleukin-1 beta expressing long-term hematopoietic stem cells showed also detectable levels of interleukin-1 receptor antagonist. And finally, within the stromal compartment, we found cells expressing one or the other cytokine or both of them. And single-cell RNA sequencing was able to dissect that most of the cells expressing interleukin-1 receptor antagonist only were mesenchymal stromal cells. But arteriolar cells were able to express either interleukin-1 beta or both, with a small fraction of cells expressing interleukin-1 receptor antagonist only. So taken together, this means that interleukin-1 receptor antagonist buffers mainly paracrine responses in the bone marrow, as shown by mesenchymal stromal cells, small fractions of arteriolar cells, but also neutrophils and monocytes. And the autocrine responses, which are mainly shown by cells expressing both cytokines, are rare. And they are found mainly within the long-term hematopoietic stem cell compartment, also within the arteriolar compartment, and some subsets of neutrophils like the most mature G4. So to then study the role of interleukin-1 receptor antagonist in a model of pre-leukemic myelopoiesis, we use the inducible and conditional Arranz G12D model. And as expected, because of the signaling pathway, we found increased levels of interleukin-1 beta in the bone marrow of these mice, but also lower levels of the antagonist. In this slide, again, you can see the main cell subsets contributing to interleukin-1 beta and the antagonist in the bone marrow of diseased mice versus controls. So in this case, the high expression of interleukin-1 beta can be explained on the grounds of the expanded multipotent progenitor compartment and myeloid compartment, together with an engagement of IL-1 beta positive feedback loop within hematopoietic stem and progenitor cells. Conversely, the low levels of the antagonist, they can be explained on the grounds of the reduced mesenchymal stromal cell compartment, together with a reduction in the expression of this gene in mesenchymal stromal cells and in multipotent progenitors. And these low levels of expression of the antagonist, they may be pathogenic, because when we delete the antagonist in either the hematopoietic compartment or the stromal compartment, there is an aggravation of the myelopoiesis. Conversely, when we treat the mice with the antagonist, there is a protection against the disease. So, in conclusion, what we show is that Hematopoietic stem cell differentiation is in fact controlled by balanced levels of interleukin-1 beta and the antagonist, and the adequate levels of the antagonist balance myelopoiesis and enable B cell development. We further suggest that a loss of repression of interleukin-1 beta signaling pathway may underlie pre-leukemic lesion and acute myeloid leukemia progression. So with this, I would like to thank the members of my research group, present and past, especially the main authors of this work, Alicia Villatoro and Vincent Cuminetti. I would also like to thank our collaborators here and many of them at CNIC in Madrid, especially the group of Andres Hidalgo, Ana Lopazo, and Fatima Sanchez Cabo. Also, a big thank you to the institutions that have hosted us during the development of this work. We had an issue with the animal facility that was closed for many years, and so we were hosted by the NCMM and by UIO. We're very thankful for that. And finally, our funding sources. Thank you very much for your attention. I'm happy to take questions.